This is a production of Cornell University.
So I'm going to do my best to use the mouse as a pointer here so that everyone can see in both locations. It's my great pleasure to give an overview of our research. I'm going to touch on a number of projects that we've got going, but there are a number of projects that I don't have time to get to. And I'm going to break up my talk into 4 sections here. So here's an overview of what I'm going to talk about, starting with the most applied and ending with the most basic aspects of our research. We'll start by talking about the overall agronomic system of growing shrub willow as a bioenergy crop, give an overview of our breeding program and some of the unique aspects of breeding willow. I'll talk about our emerging resources for mapping in willow, both QTL mapping and association mapping. And then get to a project that's funded by the Department of Energy, their feedstock genomics program, where we're trying to look at the genomic basis for heterosis in triploid hybrids of willow. So this is the typical land that we target to grow willow on. Willow as a pioneer species will do well on marginal ag land, and it is a crop that we aim to avoid the conflict between food and fuel by targeting underutilized or abandoned ag land. So this is a site at Vernon Verona Sherrill High School near Rome, New York, that is typical of a lot of the abandoned or underutilized ag land in the Northeast and Upper Midwest. With willow, we can convert that marginally profitable land back to more conventional agriculture, although very low input and very low labor requiring agriculture. So we planted willow at the high school here in 2010. I'm sorry, in spring of 2009, and this is what it looked like by October of 2010. At that high school, they have a very active FFA program where they make maple syrup, and they're actually harvesting the willow to burn in their evaporators while they make maple syrup. So here's an overview of the willow agronomic system. We are aiming to develop conservation tillage as a method of site preparation. You see here that the first thing we have to do is kill the existing vegetation. And as a part of that, for both weed management and nutrient management, Eric Fabio in my lab is doing a lot of work on first planting cover crops as a part of that site preparation. But we've also been working with Agronetics, a company in Rochester, to use GPS guidance for zone tillage, which is what you see here, followed up by GPS-guided planting using one of these mechanical planters. This one was built in Denmark. So we're planting dormant cuttings, stem cuttings. Here, these are whips that are about 7 or 8 feet long that the planter will cut into 8-inch pieces and push into the ground as it moves along. We plant in a double row system with 30 inches between the rows, with either a 5 or a 6 foot alley between those double rows, with 2 feet between the plants, giving us a planting density of about 6,000 plants per acre. Those plants will, those cuttings will root fairly readily and push buds to make new shoots that will grow about 5 or 6 feet tall by the end of that establishment year. And we take advantage of the natural biology of willow by cutting them back or coppicing them, which then stimulates vigorous regrowth in the start of that second year. So while we discard the biomass that's produced in that establishment year, Once those plants are established, they will vigorously resprout after each cutback. By the end of that first year after coppice, or the second year of production on that site, we typically have growth of 12 to 15 feet tall, and our normal production cycle would go on a 3-year harvest rotation, by which time the plants are 20 to 25 feet tall. We harvest in the wintertime. Typically we're planting on poorly drained sites that are wet. So in the winter it is an advantage to have the ground frozen. It's a time when forage harvesters like this New Holland forage harvester are not typically being used. So that harvesting operation can switch off their corn harvesting head and put on a willow cutter head and continue to use that machinery through the winter. It's a time when we need chips for heating, and obviously it's a time when the plants are dormant, so it's a good time to cut them back. And you see that all the leaves have fallen and returned back to the soil, recycling those nutrients in the leaves back to the soil. Those wood chips then would be collected, blown into a truck, taken to the conversion facility. Here you see a whole truck dumper at a power plant just north of Rome, New York, where they're piling up chips to load into their boiler. Again, that following spring, the plants would vigorously resprout, starting a new growth cycle, a new 3-year harvest cycle. We modeled the economics based on 7 of these 3-year harvest cycles, but there's good evidence from Sweden that yield is maintained for at least 10 harvest cycles. So there's really no end in sight in the productivity of a willow plantation once it's established. That biomass can be used in a number of different conversion processes. Here in New York State, we have 3 wood-fired power plants, and willow is more than adequate fuel for those power plants. There is an emerging market for feedstocks for liquid transportation fuels, and in fact the US government has set some very aggressive targets to produce 20 billion gallons of advanced renewable biofuels by 2022. We're nowhere close to getting to that point because the technology for that conversion is lagging behind and it is quite expensive at this point compared to petroleum. Another viable market that we have in the Northeast is to use wood for renewable heating. And this is a small-scale boiler that we've installed here in Geneva to heat 2 of our buildings entirely fueled by willow. Willow can also readily be pelletized and used in pellet stoves or pellet boilers. We've done a fair amount of work to look at the wood composition of willow. That composition will be critical for many of these liquid biofuels conversion methods. It's not so critical, but even still, thermochemical conversion, combustion, or thermochemical conversion to liquid transportation fuels will also have its own optimal biomass composition, and we see quite a bit of variation in biomass composition among our different varieties in our breeding program. So unfortunately, that's about all that I have time to talk about in terms of biomass composition, but it is a critical trait for these different conversion systems. Just to emphasize that willow is a viable commercial crop and that the government is invested in developing willow as a crop. In 2011, the USDA initiated a program to provide planting subsidies for willow in northern New York specifically. This overall program is called the Biomass Crop Assistance Program, and our area in New York, among these 9 counties, I believe it is specifically to pay 75% of the establishment costs to plant willow, provide an annual land payment, and to link the harvest of that willow with conversion in local power plant facilities, these 3 wood-fired power plants that I talked about. One at Fort Drum called Black River, this one just north of Rome, the Lionsdale power plant, and one up near Messina, the Chateauguay power plant. Those power plants are then paying a contract price set by the BCAP program. So we have about 1,200 acres, about 500 hectares or so of shrub willow in commercial production in New York State. There's about 20,000 acres in production in Sweden, in Denmark, in England, growing production in Poland and Ukraine, Hungary. So it is a viable commercial crop being developed for renewable energy. So that sort of summarizes our production system, our agronomics. Again, just want to point out that there are significant barriers to adoption of willow, including the high cost of establishment. The cost of cutting production is a limiting factor compared to seed production, such as for switchgrass. It is a system that needs specialized planting and harvesting equipment, most of which is built in Europe. New Holland has invested in developing their own willow cutter head that is matched with their self-propelled forage harvesters. But still, there is a lack of commercially available planting and harvesting equipment. The key point that leads into the next section of my talk is that the economics of biomass production of willow is dramatically improved when we get increased yields per acre, as you can imagine. So breeding for improved yield, for more stable production across marginal sites is a critical factor for the long-term adoption of willow as a renewable energy source. So I'm going to move into talking about our willow breeding program. First of all, establishing our goals. First goal, well, the first 3 goals are all yield. As I just mentioned, fitting that into the economics of the system, the more you yield per acre, the more we will encourage adoption. It is a perennial crop that will stay in the ground for as long as 30 years. So long-term durable pest and disease resistance is critical. As I mentioned, wood composition is a critical aspect for matching feedstocks to conversion technology. And we also always need to be conscious of the cost of cutting production and stool form and plant form, crown form, branching, for example, is a critical aspect for efficient cutting production as well as harvesting efficiency. We're now moving into looking at aspects of stress tolerance as well for particularly marginal sites, and I'll show you that one type of site we're very interested in is reclaimed mine land in Pennsylvania and West Virginia. So keeping all of these goals in mind, an underlying premise that we work with is that we want to maintain a genetically diverse suite of pedigrees among our commercially deployed cultivars. Since we are planting clonal material, we always want to maintain a high level of genetic diversity in the field to hedge against any individual cultivar failure due to pest and disease susceptibility. So our overall approach and strategy in breeding willow is to rely on species hybridization and the hybrid vigor or heterosis that we observe in species hybrids. So here I've got pictures of 3 people who have been critical over the years in actually doing the controlled pollinations and the crosses that we've done. Starting in 1998 and 99, Rich Kopp, who was one of my PhD students at SUNY ESF, really developed our techniques for extracting pollen, doing controlled pollinations in the greenhouse, did our initial hybridizations, and many of our current commercial cultivars are genotypes that Rich bred, many of which we've now patented. Kim Cameron took over for Rich and did a number of very successful crosses in 2002 through 2005, which I'm going to talk about as well. Working as a postdoc in my lab, first at ESF and then helping me move here to Cornell. And more recently, Sam Nopka, who was a Hobart student, helped out in the greenhouse over the last 2 years, helping to manage and maintain thousands of seedlings in the greenhouse. He was a real asset to us. So one of the features of Salix, and I'm going to distinguish Salix from Populus, the two main species in the Salicaceae. One of the features of Salix is the tremendous species diversity that we see in that genus. There are over 300 species of Salix, while in Populus there are only, I think, 34 different species. And they range from dwarf, sprawling alpine shrubs to, the weeping willow or black willow trees that we're familiar with. We work exclusively with species in the subgenus Vitrix, which are primarily shrub form willow. They grow with multiple small diameter stems, never really achieving a single stem like the tree form willows do. And in particular, we're working primarily within the section helix and making crosses with the section Vimin. But you see there is great potential for species hybridization here. Another feature that's unique about Salix, especially compared to Populus, is the abundance of polyploidy across the genus. And in this particular scheme of species that we work with in our breeding program, you'll see that Salix maibiana from Japan is tetraploid, a natural tetraploid, and Salix dasyclados, very common in the European breeding programs, is hexaploid, while all the other species here are diploids. I'm going to talk about some of our genomics, and we focused in on Salix purpurea as a reference genome for our initial genome sequencing efforts. It is a central species in our breeding program. It is a consistent diploid, but you can see that it is a good species to hybridize with many of the other species in section helix and also across sections here with members of Vimin. I'm also going to talk about, in our work on triploids, crosses between Salix purpurea and Salix maibiana, as well as crosses between Salix viminalis, which is the main species in the European willow breeding programs, and Salix maibiana. As you can imagine, these crosses between tetraploids and diploids will generate triploid progeny, and that will be an ongoing theme here through the rest of my talk. So since 1998, this is a list of all of the different species combinations that we've been able to produce over the years. I've underlined the species that are triploids, so you see a significant number of those progeny are triploid. But we have the capability to combine multiple species here, and our long-term strategy is for overall improvement of diploid species independently of tetraploids to make that final F1 hybridization between the diploid and the tetraploid, generating triploid progeny. Since I moved to Cornell in 2009, we've produced about 10,000 seedling progeny, representing our next round of potential cultivars for selection. So up till now, we've largely been relying on phenotypic selection, and the other willow breeding programs, the main ones being in England and in Sweden as well, have been relying on phenotypic selection. And this is our overall scheme that we've been using. First, to do those controlled pollinations in the greenhouse, producing seed that we immediately sow and get started in the greenhouse. In midsummer, we transplant those out to the field. So here we're talking about thousands of progeny. outplanted to the field here, as you see we're doing in our nursery beds here in Geneva. And then typically we would evaluate seedlings within family rows in those nursery beds and make a selection on the order of 10 to 15% within the best families, and then propagate those to replicated selection trials. And these have only been on single sites. And here we're talking about on the order of 60 to 80 genotypes that are advanced into these selection trials. Finally, from analysis of those selection trials, we select on the order of 12 to 15 genotypes to advance into regional yield trials, where it's critical to evaluate genotype by environment interactions, to look at their response to varying pest and disease pressure on different sites, their response to different soil types and climate conditions. So overall, that phenotypic selection process takes us to regional yield trials within 4 to 7 years, and our overall cycle of identifying new cultivars runs on a cycle of about 10 to 12 years. So we went through that process again using genotypes that Rich Kopp bred in 1998, 1999. Got those into yield trials that were planted from 2005 up through 2008 across New York State, and through collaboration across many parts of North America, all the way from Montreal, Quebec, out to Waseca, Minnesota, just south of Minneapolis, even in the Upper Peninsula of Michigan. So those are our target regions, the Northeast and Upper Midwest and southern parts of Canada. And you can see when we look at the yield of the top 5 genotypes in each of these trials and compare them with the foundation breeding material that we had in Syracuse originally and yield trials that were managed by my colleagues Tim Volk and Larry Abrahamson, those new cultivars from first round of breeding produced about a 30 to 35% increase in yield overall. So that's our success in one round of breeding through capturing heterosis of species hybrids. Among those cultivars that we tested in those yield trials, we have now patented 8 of them, and those are licensed by the Research Foundation of SUNY to a very active and collaborative nursery producer. Many of you know through his primary business, which is producing wine grape cuttings, Dennis Rack at AA Willow. So he now is the main source of planting stock for commercial growers in North America with production capacity of about 30 million cuttings per year. That's enough for 5,000 to 6,000 acres of willow each year. I just want to highlight again that many of these cultivars are hybrids between the diploid Salix viminalis and tetraploid Salix maibiana, or between the diploid Salix purpurea and tetraploid Salix maibiana. Okay. We have now continued to advance our breeding program, and our next round of genetic selection trials was planted here in Geneva in 2008. Again, with 75 genotypes. We harvested that the first time in 2011. And from those genotypes, bred in 2002 and 2005, and some in 2001, again, we have captured about a 15% increase in yield compared to our current commercial cultivars. So these are now, have now been advanced to regional yield trials to do that same type of genotype by environment interaction testing. But now we're getting into the yields of 17 dry metric tons per hectare per year that are really going to have an impact on the economics of the system. So I am very fortunate to be a member of a USDA-funded project to develop willow and switchgrass as bioenergy crops in the Northeast U.S. It's led by Penn State. And our contribution to this project, which we call New Bio, was to establish a new round of regional yield trials through collaboration with SUNY ESF, AA Willow and Fredonia, folks at Penn State, Marvin Hall and John Carlson, including a site on reclaimed mineland, collaboration with Steve DeFazio at West Virginia University, and with Ray Miller up in Escamaba, Michigan. So we expect that once we have the results of this new round of yield trial, we will advance at least a couple new cultivars to commercial production. Now, we recognize that our potential to capture those yield improvements is getting smaller and smaller, meaning that we need to be more precise in our selection methods. And we have recently moved to planting, to not relying so heavily on unreplicated performance in these family row trials in nursery beds, but rather propagating all of the genotypes that we produce in a family and testing them now in replicated trials. As you can imagine, these are getting very large. When you pick almost 300 genotypes, or here we have 1,100 genotypes replicated in 4 reps. I would love to do this on multiple sites, but even on one site, this is a lot of plots to go out and measure and evaluate. But that is what we have done here in 2013. Fred Gauker is my PhD student, is managing this trial, and Craig Carlson, my third PhD student, is managing this trial both here in Geneva. Okay, so to summarize our current strategy for breeding Salix hybrids is to capture heterosis through species hybridization, especially to produce triploids. That our initial selections produce some impressive yield gains, but in order to maintain those, we will need to get more precise and produce larger families with better overall precision of selection, which we think we can gain, and also gain advantages of early selection by developing genomic tools and developing marker-assisted selection. So really what we're talking about is whether we can pick a needle from a haystack, right? Whether we can pick those particular genotypes out of thousands that we can produce through breeding that have all of the traits and additive genes combined in that one single superior genotype. So I wish I could just look at the pots on the greenhouse bench and pick out that one needle out of the haystack of seedlings. But again, we can't do that by eye. We need to develop genomic tools that can do that for us. So we were very fortunate to collaborate with folks at DOE, especially Jerry Tuskin at Oak Ridge National Lab, and his relying on his connections with the Joint Genome Institute of DOE, who have invested resources to sequence Salix purpurea as a reference for Salix. So we selected one of our female clones, again a central clone in our breeding program, did entirely Illumina sequencing. That was done at JGI. The assembly was done by Haibao Tang at the Craig Venter Institute in Chris Towns' lab. And we now have released that first assembly in Phytosome version 10. This is the DOE Genomics website. So we're up to here, if you're a genome jockey, we're up to scaffolds with an L50 of 190 kb, but we still have about 9,300 scaffolds in our assembly. So basically what that means is using this short read Illumina technology, we have assembled the genome, but still it's in a large number of small pieces. Our ultimate aim is to assemble all of those sequences into chromosome scale assemblies, but that is going to take some additional sequencing work, which we are undertaking as we speak. What we have been able to do is to take advantage of this great technology developed in Ed Buckler's lab. Ed is in the USDA down in Ithaca, called Genotyping by Sequencing. This high-resolution, high-density marker system can be developed without any prior genomic sequence knowledge. It's very cheap per sample, so we can get thousands of markers for only $35 per sample. And we were very lucky to get in early on as Ed and Rob Elshire and Sharon Mitchell down in Ithaca were developing this technique to develop GBS markers for Salix purpurea, which has now helped us in developing a genetic map and improving our overall genome assembly. So what we did was to apply GBS to an F2 family, fairly large, 495 progeny of Salix purpurea, developed using our reference genome, 9406, as the original grandmother, so that we can develop, we produced, in collaboration with Steve DeFazio and Eli Rogers Melnick at West Virginia University, a high-resolution genetic map of this pedigree. We could then take those GBS markers and align them to our scaffolds in the assembly, and using the powers of the genetic mapping, could then assemble our scaffolds into chromosome scale pseudomolecule level molecules. So we have about 75 or 80% of the assembly now aligned into these 19 pseudomolecules equivalent to the 19 chromosomes of Salix. What I'm showing here in this figure is the alignment of the Salix map to the poplar genome, and here is the alignment of the Salix sequence assembly scaffolds aligned to the genetic map. Some things that we've learned, and we continue to analyze the genome assembly, is that there are some major chromosome rearrangements between Salix and Populus. Big one here is between chromosome 1 and 16, there's a fusion between Poplar and Salix. A major chunk of Poplar chromosome 1 is fused with Salix chromosome 16. We also see some important translocations, and I'm going to tip my hand here and say that we have been mapping the sex determination locus, and actually that sex determination locus sits in a region that is translocated from Poplar chromosome 19 to Salix chromosome 15. So we're learning some important differences between Poplar and Willow. Having the genome assembly here, there's a lot more analysis that we will be doing in the near future. At JGI, they also sequenced some of the expressed genes so that we could develop a high-quality annotation of all of the genes in the genome, doing RNA-seq from 8 different tissue libraries made from 5 different tissues here. Basically, what we see is that there are slightly fewer genes expressed in Salix than there are in Poplar, but still we have a pretty good library or annotation of the 38,000 or so genes expressed in Willow. Okay, the next step is to use that genetic map, those GBS markers, and the genome sequence to develop mapping populations so that we can relate phenotype to genotype, so that we can start to work towards marker-assisted selection. And we are using the complementary approaches of both QTL mapping using biparental populations with association mapping using a diverse collection of Salix purpurea. So we're now going back to that same F2 family, which has a pretty large number of progeny. And Craig Carlson here has planted that out in a replicated trial here in Geneva so that we can start to characterize traits of importance for biomass production, for willow biology, and map them into this high-resolution biparental mapping population. We've done that on some of the simple traits already. Since willow is dioecious, we have been able to map the locus controlling sex in willow. As I mentioned before, it maps to a single spot on chromosome 15, and in fact, we see that Salix purpurea, and perhaps all Salix, are using a ZW sex determination system. That means that the female is the heterogametic sex, the male is ZZ, while at least Populus trichocarpa uses an XY sex determination system. So again, there are some interesting contrasts there between poplar and Salix, and in the overall evolution of dioecious, and whether that XY and ZW system can flip-flop back and forth, or at what point in the divergence between poplar and Salix did it flip-flop. That's just one example of the types of traits that we can map using this powerful system. Now, Fred Gauker has been working to develop association mapping, or a genome-wide association study, or GWAS, using the collection of Salix purpurea that we have assembled from across the Northeast. So we have, it's a relatively small association mapping population, only 112 genotypes, but we have planted that on replicated trials here in Geneva, out at the Clairol station in Portland, and in collaboration with Steve DeFazio down in Morgantown, West Virginia. We have fully genotyped that collection with GBS, and for the last 2 years, Fred has been working on phenotyping those populations with some of the key traits, again, that we're interested in for biomass production, including, as you can imagine, stem height, stem diameters, stem numbers. We're very interested in wood density. We use high-resolution thermogravimetric analysis to determine the composition of cellulose, hemicellulose, lignin, and ash in the wood. And we've also been able to measure leaf area, use a SPAD meter to measure leaf nitrogen, and a pyrometer to measure stomatal conductance. We've also developed some techniques to look at overall crown form and growth form. Also looking at pest and disease resistance, which aren't on these, which isn't on this list. Again, some of the initial hits that Fred has been able to map, and he's actively working on mapping all of those traits right now. Again, a simple one is to look at sex determination. And the mapping in the association population matches that we see in the biparental QTL population, pointing to a single major locus on chromosome 15. So that highlights how we can use these two different mapping populations to complement one another and to confirm the hits that we're getting in both types of mapping. Some other hits that Fred has come up with here, we see a significant hit for variation in stem height. So as we look at these Manhattan plots, each of these different colors represents the 19 different chromosomes in Salix, and each dot represents a SNP or a marker on that chromosome. As it gets higher in this plot, represents a higher level of significance for the correlation between variation in that trait and that particular spot on the genome, or SNP. So looking at hits for stem height here, he has some SNPs that are significant and map to this very specific region of the genome, which we can map using, I think this was done with about 25,000 GBS markers, mapping those SNPs between a glycosyl hydrolase and a cellulase, which we now need to follow up on and see how they are involved in regulating stem height. Another hit for hemicellulose content quantified by TGA. Here we see significant SNPs here in a sugar nucleotide epimerase, potentially involved again in sugar nucleotide conversions during cell wall biosynthesis. But this gives us some targets, some candidate genes that we can follow up on, look at their patterns of expression, look at the variation in alleles in our association population, and to see how those different alleles are influencing the traits involved. Okay, so to summarize, we have a pretty good genome assembly in place right now, produced through good collaboration with the Joint Genome Institute of DOE. We continue to advance those genomic resources. We have developed both QTL and association mapping populations that can complement each other. The big issue, the most expensive part now is phenotyping those populations with thousands of plots out in the field. But we have great potential for identifying good candidates for marker-assisted selection. Now, the final part of my talk is that I think that Salix also represents a unique system for the study of heterosis, and that having these genomic resources gives us the potential to probe some of the genomic basis for heterosis in this highly heterozygous species which has undergone no domestication up to this point, and also displays heterosis in across ploidy levels, or in triploids. Okay, so I'm going to take you back to this 2008 selection trial, which had 75 genotypes. And if we look at the yield, the mean yield of those 75 genotypes in that trial, and look at the ploidy, the vast majority of the upper 50th percentile of genotypes in that trial were triploid. And the overall yield of the triploids was significantly and dramatically greater than the yield of the diploids in that trial. The yield of the tetraploids is not significantly different, but when we look at some of the other traits like stem number or aspects of biomass composition, we do see significant differences between the triploids and the tetraploids. The bottom line here, again, is that we are seeing dramatically better performance in triploids compared to diploids. So we want to ask, what is the genomic basis for that heterosis that we're capturing? So just to sort of define what we mean by heterosis, the classic example is in maize, where if you cross one inbred line with another inbred line, you see that the progeny display either mid-parent heterosis, meaning that their mean performance is better than the average performance of the two parents, or you may see better parent heterosis, meaning that the progeny are better than even the best, the better of the two parents, right? And there are some classic models to explain how that heterosis is occurring, whether it's occurring through a dominance model or through an overdominance model. The dominance model meaning that there are complementary additive alleles which come together in the hybrid to make that hybrid even better than its parents, or in the other model, the overdominance model, where the presence of a heterozygote, even at a single gene, can confer superior performance to that hybrid over its two parents. The main difference being that in the maize example, those are inbreds, highly homozygous inbred lines that have been bred for decades. Here we're seeing heterosis when we cross two wild accessions that are highly heterozygous, and when we cross a diploid with a tetraploid to get a triploid. Right? So the approach that we're taking is to look at the patterns of gene expression in the triploid progeny. And to what extent is the inheritance of gene expression in the hybrid progeny non-additive, meaning different from the mid-parent average? Can we identify allele-specific expression inherited from one or the other parents? And the obvious question would be, is there a dosage effect in those triploid progeny that are getting 2 copies from the tetraploid parent and 1 copy from the diploid parent? Finally, are those differentially expressed genes responsible for heterosis? So can we correlate the patterns of gene expression that we see with phenotype in the field or in the greenhouse? So as we look at quantifying patterns of gene expression, this is a scheme that we use. First of all, sort of the default is when gene expression equals in parent 1 is equivalent to that in parent 2. Here, parent 1 will always be the maternal parent. So when there's no differential expression and the progeny gene expression is at that same level, we classify that as additive. That's not so interesting to us. The more interesting case is when there is differential gene expression between the two parents. Here the mother has higher expression than the father. Here the mother has lower expression than the father. If the progeny expression hits the midpoint between the parental levels of expression, again, we would call that additive. If the progeny expression is equivalent to the high parent or the low parent, then that's an example of dominance. Or if it is below the low parent or above the high parent, again, that is a non-additive pattern of expression, an example of underdominance or overdominance. So in the cases where we see differential expression between our two parents, we will classify the expression in the progeny according to these 5 categories. Okay? In order to test, to look at differential patterns of expression, we've developed these 8 families. First of all, an F1 family of Salix purpurea, that same F2 family of Salix purpurea that I've talked about, and then reciprocal crosses between purpurea and viminalis, here, purpurea as the mother or viminalis as the mother, and then triploid families representing crosses between purpurea and myobiana or between viminalis and myobiana. And we have planted typically 100 progeny from each of these families in the field. And the experiment that I'm going to describe to you was done by selecting 10 random progeny from 6 of these 8 families, growing those in the greenhouse, and then we extracted RNA to use for RNA-seq. Okay? We extracted extracted RNA from 2 different tissues, either from shoot tips or from stem internodes, and mapped those reads back to our genome assembly, did statistics to look for differential patterns of expression, and we will continue on. Unfortunately, I don't have time to talk about this today, but use some additional techniques to look at allele-specific expression. Okay, just to quickly go through some of the main conclusions. The first one being that triploids express more genes than diploids. So when we look at the total counts of genes that are expressed in the triploid progeny, we see that there are thousands of genes thousands of additional genes expressed in the triploids than in the diploids. So that may be one clue right there. Interestingly, the overall levels of expression tend to be lower in the progeny than they are in the parents. Now, when we cluster the patterns of gene expression between progeny and the mid-parent values here, the first level of clustering that we see is distinguishing progeny from those mid-parent values. So the greatest level of difference in expression is between progeny and parents. The next level of clustering is at the level of tissue. And finally, sort of the bottom level of clustering is at the level of species. So it's interesting that the greatest difference in the gene expression patterns that we're seeing are differences between progeny and parents, even greater than differences between species. To put that another way, we see greater differential gene expression between the mid-parent and the progeny than we see between the two parents, which are different species, right? So here, when we compare one parent to another, male to female, you see that most of the dots are fairly close to the one-to-one line. But when we compare progeny to mid-parent, now we have a much broader scattering of spots representing greater differential gene expression between progeny and parent. Okay, the next thing was to quantify among those genes which are differentially expressed between the two parents. Okay, so statistically, which genes for which parent 1 is not equal to parent 2. And as you can see here, the triploids and the interspecific diploids have far more genes showing differential expression between the two parents than our Salix purpurea families. So of about 35,000 genes here, as many as 7,000 genes are differentially expressed between the two parents. Okay? So now, for those differentially expressed genes, and there are more in the shoot tip than there are in the internode, for those differentially expressed genes, we fit them into those 5 categories. Are they maternally dominant, paternally dominant, overdominant or underdominant, or are they in that sort of default additive case where they hit the mid-parent mean? And the interesting thing here is that those genes that show additive expression are very, very few. In fact, the vast majority are showing highly maternal dominant patterns of expression. So again, that may be a clue to the heterosis that we're seeing in species hybrids in Salix. We see more in shoot tip than we do in internode. Finally, what we want to go on to look at is can we actually look at the allele coming from the mother and the expression of the mother's allele compared to the father's allele. We needed to identify SNPs or sequence differences between those two parents. So we have now resequenced all of our parents in this experiment. And we see quite a few differences, SNPs, that we can rely on to give us as many as 18,000 SNPs so we can start to sort out allele-specific patterns of expression between mother and father. Okay, so to summarize this final experiment that I described, there is greater differential gene expression in triploids than in our Salix purpurea diploids. That the gene expression levels in the progeny show a strong pattern of maternal parent dominance, which is very interesting. The next step that we need to move on to is to start to phenotype individuals in the field and in the greenhouse and correlate the patterns of gene expression and perhaps expression of individual genes that are differentially expressed with the heterotic phenotypes that we can quantify in the greenhouse or in the field. So that's all I have time to talk about today. I want to express my sincere thanks for all the folks who have worked in my lab over the years, my 3 current PhD students, Fred, Eric, and Craig, and Kurt, my technician, Michelle Sarapelia, who was my graduate student and then postdoc here in Geneva, who did a lot of work to develop our methods of analysis of wood composition, and my collaborators, Jerry Tuscan at Oak Ridge, Steve DeFazio at West Virginia, Chris Town at Craig Venter Institute. I mentioned my collaborators at Michigan State, Penn State, and SUNY ESF, as well as all of the crews that we've had here working with us in the summer, primarily here in Geneva. So I will stop there. Thank you for your attention, and I'd be happy to answer any questions, including those from Ithaca, if you unmute your microphone. So thanks.
Gennaro.
Quick question. Do you think the differential expression that you see in the mother genome is related to the connection with the plastid genomes that are inherited, and therefore photosynthesis and everything else, all the other machinery? Do you think that is reasonable, that they communicate better because they've evolved together?
Yeah, so just to repeat Gennaro's question, the question is whether the strong maternal dominance of expression that we're seeing may be due to the influence of the chloroplast genome, which is inherited from the mother. I think that's an excellent hypothesis that we can test, and Craig, I know, is very interested in testing that hypothesis and has looked at the expression of chloroplast genes also in our RNA-seq data. The other question is whether there is some epigenetic imprinting involved in this maternal dominant expression. And the maternal dominance, let me reemphasize, does not matter which species, right? So whether Salix viminalis is the mother or Salix purpurea is the mother, it is maternal dominant, it is not species specific. Bruce?
Larry, you know, in some species like potato, when you make a cross between a tetraploid and a diploid, you get a pretty high frequency of tetraploids due to unreduced or diploid gametes. So I'm wondering, out of your triploids, what you've done to confirm that they're triploids, and I'm wondering whether diploid gametes occur among Salix species.
Yeah, so Bruce's question is how detailed have we gotten to confirm that they're triploids? At this point, we've only done flow cytometry. We've done repeated flow cytometry because that technique can be a little inconsistent from run to run. We have not ruled out that there could be different levels of aneuploidy. I think we could go back and look specifically through our GBS data to look for GBS markers that might indicate aneuploidy of a particular chromosome. Yes, we do see unreduced gametes in Salix because sometimes we are able to get progeny from our triploids. So that's probably the best evidence that I can give you that there is some shaky control of chromosome number in gamete production.
No. You show that there's some native polyploid, tetraploid, and hexaploid. I just wondering, are those the allo or it's auto polyploid? And if it's auto, again, that will be question related to what Bruce just mentioned earlier, the gametes and reduction. You know, how that might complicate the breeding system.
Yeah, I don't know how detailed anyone has characterized, so the question is whether the polyploids are allo or auto. I'm not aware of whether anyone has done any detailed descriptions of those, but I would assume that they are auto polyploids. Yeah. Thomas, and then maybe we should go to Ithaca.
Okay, so I'll ask for production. So you said that having several genotypes in a field is superior to a single one. But it strikes me, if you say you had 3 genotypes in a field and one of them got wiped out, now you're in a field with only 2/3 of a crop, which can't possibly be economical. Isn't it better to just wipe out the whole thing and start over if you get one of your genotypes with a fatal flaw?
Okay, so so if you have picked the one best yielding cultivar and have planted all of your fields with that one, and it gets wiped out, then you have no yield.
Right.
If you have planted 3 and you planted them in blocks, or maybe this field in one, this field in another, then yeah, your one field is wiped out, you can replant it. Or that one block is wiped out, you can replant it. The standard in Ireland now, and in England, I think, is to plant in random mixtures. And over time, the superior genotypes win out, the rust-susceptible genotypes diminish, and you end up with a lower number of plants per acre. But what we found is that that does not severely impact overall yield.
How much percent plant loss can you handle and have it compensate?
I think you can go down probably 30% in stand density and still realize comparable yield. Any questions from Ithaca?
Yes, you indicated that there are going to be 7 harvests commercially, so the first question is any fertilizer put on that at all, because you're removing a lot of nutrients from the soil? And as a corollary of that, it would seem to me that over the long term, the plant's ability to take up nutrients would be a considerable factor in the total yield. But you never mentioned that, at least I don't think so. So I'd like to know whether there's any plans to look at your progeny for the ability to take up nutrients.
Yeah, thank you, Peter. That's an excellent question. And it's one of the aspects of our research that I had to drop out from this seminar, but is the main topic of Eric Fabio's research. So he has done fertilizer trials using some of our current commercial cultivars in the greenhouse, has been looking at the impact of nutrient management by cover crops on early establishment of willows in the field, and again has been analyzing yields from across now 24 different trials to correlate soil nutrient levels with yield and growth parameters. There are conflicting data in the literature, some papers which say there's very little influence of fertilizer. I believe that there is an influence of fertilizer, especially in the early harvest cycles, but that it seems fairly likely that after a certain amount of time, a cycling of nutrients will be established in willow plantations so that long-term fertilizer application may not be necessary. Any other?
Okay, I think that's all the questions we have here.
Okay.
Thank you.
One more question.
Willows mycorrhizal.
Jay is asking if willows are mycorrhizal, and yes, they are. And they are rather promiscuous. They can form associations with multiple different taxa of mycorrhizal. That's a very interesting question. and again, ties in with the need for fertilizer, but it's one that needs a lot more attention. Okay, one more.
What type of marker you are using for your F2 population? Like whether you are using all kinds of marker like intercross, pseudo-test cross, and how many you have?
Yeah, so we have used both pseudo-test cross, male parent backcross markers and female parent backcross markers, as well as intercross markers. I think in that initial analysis we had about 8,300 markers, and I think about 3,300 of them were intercross markers, and something like 22,000 or 2,200 were pseudo-test cross markers. Yeah. Okay. Thanks very much.
This has been a production of Cornell University, on the web at cornell.edu.